Hi everyone, welcome back to this week lecture. Apologize because last Tuesday I was not able to complete this recording, but today I'm gonna do it. So there's two parts for today's lecture. First one is I'm gonna continue. I was supposed to do this on Tuesday, but I really forgot about it. I just remember that previous week's lecture was not completed. So um, I'm gonna go through a little bit on part one, which is from protein to amino acid sequence analysis, because last time we did not finish all 5 steps. So today we're gonna look at step number 4, which is degradation from the N terminus, and step number 5, which is the degradation from the C terminus. And then following that, we'll look at a little bit of introduction to lipids. What are lipids? Different types of lipids. The what do you call it? The categories or the subsets of lipids.
Alright, so now you know your sequence, but again, if your protein is very, very huge, um, even bigger than this one is quite short, right? If your protein is very, very huge, so how do you know which one is, well, even for this one, you don't know which one is C, which one is N, right? Because for all you know, C can be from that side, and N is from this side. You don't know until you verify. So to verify which one is the N terminal, which one is the C terminal, we'll move to step number 4 and 5. Okay, step number 4 is a method to identify the N-terminal amino acid. So the first amino acid attached to the N terminus of a polypeptide is treated with PITC, okay, phenyl isothiocyanate, a more common known as Edmonds reagent. So Edmonds is N-terminal cleavage. So what does it do? So the Edmonds reagent, or PITC, react with the N-terminal amino group, and the resulted in thioazolinone derivatives. So this thioazolinone derivatives is cleaved from the polypeptide under mild acidic condition, leaving behind a polypeptide with one less amino acid. So it's very specific to N-terminal only. Okay, so this technique, this is just the reaction mechanism. You can have a look at it. But I'm not gonna touch about it. You can just study it on its own. You can see electron pushing, electron movement, and presence of base and acid and whatnot. Okay, so what happened with the thioazolinone is that in the presence of valylic acid, it forms a more stable PTH structure, like shown on the bottom right here. Since each amino acid has a different side chain, each amino acid forms a different PTH amino acid. In particular, PTH amino acid can be identified by chromatography using known standards. So it's already been developed. So it's similar case as previously, where you have a peak of standards, and then you run your PTH amino acid, then you know that okay, this PTH corresponds to X amino acid. So then you know your polypeptide chain has an X amino acid as the first amino acid at the N terminal. So you can actually use, or you can actually automate this system and digest one amino acid at a time, and finally you can know the exact sequence of the polypeptide. But the disadvantage is, if you were to use Edmond degradation to identify a protein which is very, very huge, insulin is still okay, it's just 21 amino acid, but for a protein that has 200 or 400 amino acids, doing this will take you months. Okay, so that's why the technology for partial digestion using enzymes are developed. Okay, so to ease up the method of identification. Alright, so this is a representation on what Edmond degradation do. So that's your original sequence. Okay, you first off, you cleave off the cysteine residues, and then you analyze this. You know that it's cysteine, that's good. And then you cleave off the second amino acid, you analyze it, then it's good again, and then you do it again and again and again and again and again until you finally know that this is your original sequence. Okay, as simple as that. And I think that's it for element degradation. Now we move to Sanger degradation. So it's opposite. It's the opposite. C-terminal amino acid can be identified using carboxy peptidase hydrolysis of one C-terminal peptide bond. And there's two types of carboxy peptidase, which is A and B. And again, element degradation is more on a chemical approach, and Sanger degradation is more on the C-terminal approach. It cleaves off the C-terminal amino acid as long as it's not arginine and lysine, and B is only if it's arginine and lysine. Again, they are either attracted to a cation or not attracted to cation. Okay, I will need to look at again for Sanger. I think Sanger degradation is something else. This is not called as Sanger. I'll have a look and confirm it with you. Okay, so imagine this is the structure of your peptide bond, where you have your N-terminal there and C-terminal here. This is the site where the carboxy peptidase will cleave. Okay, since it's not a non-specific enzyme, okay, again, if you looked at the amino acid sequence, it's only if arginine and lysine, and the other one is non-specific to arginine and lysine. So you have another 18 amino acid where it can cleave off. Okay, so it's non-specific enzyme. It's specific to protein, but it's non-specific to individual amino acid. So the rate of hydrolysis differs from one amino acid to another. So it's still useful, but not as much as element degradation, because imagine if it cleaves off glycine in 0.1 picosecond. It might cleave alanine in 0.5 picosecond. So if you put it in the reaction mixture together, or you have a sequence where you have alanine and arginine, you cannot compare this with a sequence where you have a glycine and alanine. So if you put these two sequence into separate a beaker, and then you put your carboxy exopeptidase, you'll get the same reaction time, same product within 1 second. So how can you determine whether which one is at the C-terminal? So you can't. So that's why it says it's still useful, but not as useful as element degradation, because element degradation is a chemically induced, so you can actually control the reaction very, very well. So during analysis, the appearance of free amino acid is monitored. In theory, the amino acid whose concentration increases first should be the C terminus, and the next amino acid that appears should be the second residue from the end. But in practice, however, different amino acid, because it has a different reaction rate, so it makes it difficult to determine which one of the amino acid is actually the first and the second residue. So that's a disadvantage. So compared to element degradation, where you can actually cleave off from the left hand side one at a time, for the C-terminal identification, you can only cleave up one or probably two, and then that's it, because this no longer works. Okay, because the rate are very, very different. And also because you have arginine here, okay, remember, if you use the A, it will stop there. So now you only know you have a glycine and leucine, but then again, you don't know which one is the first amino acid. Okay, so what's missing here? So what I've illustrated are the methods, or some of the methods that you can use to identify a sequence of an amino acid or protein. But it does not mean that if you have an unknown protein, you can simply follow these steps to identify the protein sequence. So you can, but it's not always true. So sometimes you get it right, sometimes you get it wrong. So a partial method is required, but it's not covered in this lecture, since it's out of the scope of this course. So there are a few other methods, partial methods that I'm not gonna talk about. And if you are still interested, if you want to work with peptides and protein in the future, you can always contact me. You have my contact details. Okay, so in summary, you have this hydrochloric acid hydrolysis or acid hydrolysis. You know a little bit about trypsin, chymotrypsin. You know a little bit about cyanogen bromide, the rate of mechanism and specificity to methionine and cysteine modified structures. We have, a little bit about Edmond degradation, and fortunately this is the correct spelling. It's not Edmond, it's Edmond. So I apologize, but I'm not going to redo this lecture. It's taking too long. And finally, a little bit about carboxypeptidases, where it cleaves off on the C terminal of the peptides. These are some of the references, and I'm gonna put some information in my own website. And I think that's it. Thank you, and the lecture length
Okay,
is...
so let's move to the first one, which is Introduction to Lipids. Second one, actually. We've already covered the first one. Second one, Introduction to Lipids. So there are a few types of lipids. So pretty much lipids can be categorized into two, at least for now, can be divided into two groups. First one is fatty acid. Fatty acid, and the second one is steroids. So why are there's only two groups? So pretty much it's because of the structure. So you will see later the basic structure for this, okay, waxes, fats, oil, and glycerophospholipids contains fatty acid. Okay, so that's why those all these four lipids are categorized into fatty acid. And then second one, we have steroids. So steroids also have its own structural motif, which is a nucleus of four conjugated carbon rings. Okay. Okay, so this is a general structure or classification. We have lipids here. It can be divided into two groups, fatty acid and steroids. And fatty acid can be further categorized, not just three. They can actually, well, depending on what journal or what literature that you are following, it can be three, as minimum as three, and as much as five. Okay, because glycerophospholipids can some says it's falls under phospholipids, okay, which is our cell membrane. And some says it's a separate entity and whatnot, but it doesn't matter. What I'm using today is I'm gonna divide fatty acid into three and steroids into two, which we will cover this on next week. Okay. Alright, so we'll look at this very, very quickly. I'm not gonna go into detail like how to synthesize and whatnot. It's more in general overview of what are fatty acids, what are waxes, triglycerides, and glycerophospholipids. Okay, so lipids are a family of biomolecule with a common property. Okay, so this is the general slide. Okay, which is soluble in many organic solvent but insoluble in water. So I would say 99% of lipids are not soluble in water. Okay, even soap, when you consider soap as a lipid, technically they are not soluble in water. Okay, because it form micelles and whatnot. But it's a totally different story. So solubility, solvation agent, it's kind of like a different realm than what we are looking for here. Okay, so lipid contents for a cell can be extracted using organic solvent, but they can never be extracted using just water. Okay, normally you will have a mix of organic solvents such as dichloromethane or diethyl and then with water, so that you can actually separate the lipid layers and the cytoplasm of a cell. Lipid comes from the Greek word lipo, meaning fat. Okay, and lipids are important feature in cell membranes, fat insoluble vitamins, and steroid hormones. So, yeah, well, pretty much. If you remember in high school, we have these four vitamins: vitamin A, D, E, and K, which is soluble in water, if I'm not mistaken, or otherwise ADEK. Yep. So ADEK is soluble in fats. Okay. The rest, like vitamin C, vitamin Bs, and whatnot, are soluble in aqueous. Okay, so added in fats. So it is still important to intake some fat, so that our body can actually enhance the absorptions of some vitamins and minerals. So some properties of lipids, as I mentioned, insoluble in water but soluble in organic solvent. You actually or potentially related to fatty acid. Okay, as you will see later. It can be used as a source of energy by animals, so especially for animals that does hibernation. Okay, so they consume a lot of lipids, seeds, fruits, and leaves, so that during their hibernation they can still process and have enough energy to sustain for their hibernation. So some animals do hibernate for as long as a year or so. Okay, and there are many more functions in the world today. If we do have time, then I'm gonna cover it in next week's lecture. But otherwise, we can just simply Google functions of lipids. Well, one of the functions that everybody uses is cooking, right? Everybody uses oil, so and everybody drive a car. So there's a lot of examples. So you can just simply Google and find a few examples. Okay, so these are the categories again. So you have lipids categorized into fatty acid and steroids. And then under fatty acids, you have waxes, triglycerides, triglycerols, triglycerides, it's the same thing, and glycerophospholipids. And under steroids, you have cholesterol and steroid hormones. So biological function of lipids. Okay, so first one, they are rich source of energy, as I mentioned, but they are very, very slowly metabolized. So that's why our body utilizes glucose primarily compared to lipids. So first is glucose, second is proteins, and then the third one is lipids. Okay, again, in terms of energy, lipids has the highest, protein is the moderate, and glucose is the lowest per molecule. But then when you consider the abundance and the biological process by which these molecules can be converted into energy, glucose is the fastest. Thus, it can provide an high instantaneous energy. Protein is a moderate and in moderate energy, and lipids has the lowest metabolism with a lowest instantaneous energy that a body or a living organism can provide or can process. Okay, so other functions of lipid. Number two, it protects and insulates internal organs. So again, I do have pictures, but I'm not going to show it. I'm afraid some of you might, be eating while listening to this lecture. So if you are interested in looking at how lipids actually protects and insulate internal organs, so by all means, please Google them. Okay, but make sure you're not eating anything, because if you're not used to biology, some of you might feel nauseated and throw up. Okay, number three, other types of lipids are found in nerve fibers and in hormones, which act as a chemical messenger. Okay, so nerve fibers, if you're looking at the structure of lipids, so you would say that lipids are actually non-conductive. But surprise, surprise, our biology developed a system whereby they use lipids as an insulator to promote electrical conductivity. If you're interested, you can come talk to me and can probably talk to you about it, or you can simply just Google and neurons, okay, and from there you can actually see some neurons, well, most neurons, they have an insulator around the what do you call it? So it's axon. I can't remember. I left biology for quite a while. Dendrites. There's this middle stem over there where it's insulated by some lipids. Okay, so that the electrical conductivity actually jumps and moves faster, which is very interesting. Biological function of lipids. Number four is provide a polyunsaturated fatty acid, which is important for our cell membrane, for our growth, cell cycle and whatnot. And number five, a major function of lipid is to build the cell membrane that separates the internal contents of a cell from its surrounding aqueous environment. Okay, so I think that one is very straightforward. So let's look at the first types of lipids, which is fatty acid. So the human body is capable of synthesizing most fatty acids from carbohydrates or other fatty acids. However, humans do not synthesize sufficient amount of fatty acid that have more than one double bond. Okay, so fatty acids, so that's lipids, and then you have an carboxylic acid group. So that's the basic structure of fatty acid. So our body can synthesize fatty acid with a one double bond, one unsaturated double bond, but we cannot synthesize more than one, at least not that we know of. Okay, one day in the future, someone might have identified, oh, we actually do have a different fatty acid, but as of today, we cannot synthesize more than one double bond fatty acid. Okay, so these fatty acids are called essential fatty acid because they must be provided by diet. Okay, so it's similar to how amino acid can be categorized into essential and non-essential, where essential means that you can only consume it or you can only get it from diet. So similarly in fatty acids, so the polyunsaturated fatty acids are the essential fatty acids. And a deficiency in essential fatty acid can cause, for example, skin dermatitis in infants, and there are so many other diseases that you can find. Okay, but fortunately for you guys, there's no assignment related to lipids and disease, so you can just complete your proteins and lipid assignment. So that's the basic structure of fatty acid. So that's the chemical structure. There's also a chemical structure via space filling model, and this one is just by what do you call it? I can't remember the name. But yeah. So those are three representation of a fatty acid structure. So fatty acid is the simplest type of lipids and are found in components of a more complex lipids. So normally it's a mix. Contains a long carbon chain attached to a carboxylic acid group at one end. So you have a carboxylic acid group over there. Although the carboxylic group is hydrophilic, the long hydrophobic carbon chain makes a long fatty acid insoluble in water. Okay, so as far as I know, okay, a fatty acid which a carbon chain with less than 3 or 4, even then they are not fully soluble in water. So normally they form micelles and whatnot. So yeah, fatty acid have even number of carbon atoms, so usually 8 to 20. But again, as a chemist, you can synthesize as low as well, 3. You don't call it fatty acid because that one is simply that, or 2. Okay, so you consider 3 to 4 is the minimum number of a fatty acid. Okay, fatty acid. Right, so again, these are some examples of fatty acids. So you have this arachidic acetic acid, stearic acid, palmitic acid, arachidonic acid. Okay, so the difference is, as you can see, comparing these two representation, a chemical representation of a fully saturated fatty acid is a linear. Okay, similarly for stearic and palmitic. But once you have our unsaturated fatty acid, you can see the structure. If you model it to the minimum energy, it's kind of like bent. Okay, or in other terms, there's a kink on the structure. Okay, on the structure. Okay, so fatty acid or FA can further be grouped into two groups: unsaturated and saturated. And unsaturated, there's another two different names. First one is monounsaturated. Mono means one. Okay, and then for more than one, we just call it polyunsaturated. It's very easy. Okay, so for that one is a polyunsaturated. Polyunsat. Okay, and these three are all unsaturated fatty acids. Saturated fatty acids fit close together in a regular pattern, thus allowing for a strong van der Waals or hydrophobic interaction between carbon chain. Requires high amount of energy to separate them to an individual unit. And as the length of carbon chain increases, so does the energy required. So as an example, if you looked at the lauric acid and palmitic acid and stearic acid, as the carbon bond lengthen, you can see the melting point is also increases. So all of these are actually solid at room temperature, okay, because of the strong energy. But again, they are only solid, but they are also a hygroscopic molecule. Okay, hygroscopic means it absorbs water, water vapor. So because again you still have this carboxylic acid group which can form a hydrogen bond with water molecule. So these molecules, even though they are solid, but if you leave it at the open, especially in the Malaysian humid environment, so they will kind of like form a squishy oil like structure with the brownish color. Okay, so in unsaturated fatty acid, the unsaturated double bonds causes kinks in the structural arrangement, thus resulted in irregular structural arrangement, weaker hydrophobic or van der Waals attraction between the carbon chain, and melting point is therefore lower than saturated fatty acid, as you can see here. Okay, so how does it form a kink? All right, so imagine if you have your saturated fatty acid. So the structure forms like in a straight manner. Okay, for something like this, which is very highly ordered, it's kind of like a solid. Remember when you learn about solid in high school, solid has a very arranged molecular structure. So similarly for lipids, for saturated lipids, they have a very ordered molecular structure. Thus, they can be kind of like a solid or a semi-solid nature. But once you have the kink in place, okay, imagine you have that and then you have a double bond somewhere over there. Okay, now you can no longer form a straight chain. Okay, so imagine and you have another kink over there that forces towards this side. So you can actually see there's a lot of spaces here and there. So there can be a lot of fluid, a lot of movement. So similarly, when you looked at the state of matter for a liquid, the structure is less orderly packed compared to a solid. So that's why normally for a unsaturated lipids, they are in a liquid form. For example, the easiest one is if you go to your kitchen, ask your mom, Mommy, where's the cooking oil? You can see this cooking oil made up of either palm oil or coconut oil, or not coconut oil, sorry, corn oil. And if you Google and try and find the structure of this molecule, you can actually see they are all unsaturated fatty acid. And some of the brand actually did mention how many percentage of saturated and unsaturated fatty acid in their oil. Okay, so again, this is a structure. A good example or the easiest example is butter compared to an oil. Okay, so they can be made from the similar structure, similar original ingredient. Okay, for example, they can be made from palm oil. Okay, and via hydrogenation, you can form butter. But palm oil, just extracted palm oil, is an oil. Okay, so because of this kink that is caused in the structure. So even though this structure is a triglycerides, but fatty acid is that. Okay, so almost similar concept, even though they are slightly different. All right, so we've looked at the general structure of fatty acid. Now we looked at a more thorough example on waxes, triglycerides, and glycerophospholipids. So basic intro, a wax is an ester of unsaturated fatty acid and a long chain of alcohol, each of approximately 10 to 30 carbon chain length. Triglycerides is triester of glycerols and fatty acid, while glycerophospholipids is the triester of two glycerols and one phospholipids. Okay, we'll look at the structure in more detail. So waxes, what are the uses? Oh, it's an habitat for bees, for example. It's the main component in a beehive. Remember the honeycomb structure, okay, where you can find baby bees in there. So these are all made of wax, bee wax. So we also use wax as water retarder. Okay, so you that's not we, nature use it. So that's a wax on the surface of, for example, apple. When you looked at apples, okay, so you normally see that apples are a bit shiny, right? Some of you, like me a long time ago, thought that, people pluck this apple and then they waxed it. But I've been to an apple farm and looked through the processing. They are actually not coated with wax at all. It's because the apple itself has a naturally occurring wax. Okay, so and that wax function as a water retarder. Okay, otherwise water will evaporate from your fruits and your fruits will be dry. So nature found its way to use wax to actually stop water from evaporating. And also on the surface of leaves, okay, so you can actually, if you pluck a leaf, you can actually feel that leaf is a bit what do you call it, silky smooth. Okay, it's because of the presence of wax. Humans, for example, uses wax as a protective coating for furnitures, for your cars, and for floors. And jojoba wax, for example, is used in making candles that we nowadays seldom use. Okay, so that's some example of waxes and their sources and uses. Okay. Triglycerides is the triester of glycerol. So triester, okay, look at that structure. It's a triester of glycerol, so that is glycerol, and these are all fatty acids. Okay, you can see that's the basic structure of fatty acid. So its highest form of energy storage in biology, okay, as fat or triacylglycerols or triglycerols. It's a high energy but slow in breaking down. So that's the general structure of a triglycerides. So again, if you looked at the chemical structure, initially you have a glycerol and you have a fatty acid. You can simply just what do you call it? Not hydrolyze, condense it, okay, conjugate it to form these ester linkages, and you can actually form a triglycerides, which is oils, wax, and whatnot. Okay, so natural fats and oils are normally contain two to three different fatty acids. So it's not the same of fatty acid. So it can be a palmitic acid plus a stearic acid in nature. Okay, so it doesn't mean that triglycerides must be from the same component. So for example, triglycerides in solid or semi-solid form at room temperature are fats in meats. Okay, so if you buy a red meat, then you normally see white fats. So they are kind of like a semi-solid. Okay, if you buy a whole milk, so it contains triglycerols. You have another example is butter and cheese, which are fully saturated fatty acid and also is a triglyceride. Another example is in oil, triglycerol in liquid form at room temperature, cooking oil of various kinds. Difference is they are polyunsaturated fatty acid. Some, for example, palm and coconut oil are solid at room temperature because they contain. Anyone can have a guess? Hopefully you do. Do I need to give an answer? Never mind. Yeah, you guys figure it out. Okay. And these are some examples of different types of fats and or oil and their melting points and the degree of polyunsaturated, monosaturated, and unsaturated fatty acid content. And as you can see, if it has, for example, that one, a very high saturated fatty acid. You can see the melting point is around or higher than room temperature. And those who are actually fully, this graph is actually a bit weird. I was expecting that to be the highest. But anyhow, yeah, well, of course, it's another thing is because the fatty acid chain length affects the melting point as well. So even though this one has the highest number of saturated fatty acid compared to butter, for example, but the fatty acid used in fatty acid in coconut and butter are totally different. So that's why for butter and you have a higher melting point, and for coconut oil you have a lower melting point. Okay, I think that's it. That's the basic introduction of fatty acid, and see you guys next week. Some references again. All right, thanks.